Myself, Muhammad Fahid Ansari, on behalf of Organizing Committee, I will take the opportunity to welcome all the participants to this session of 5 weeks National Workshop on Statistical Analysis of Biological Data. This session is also going to be very interesting, as the speaker of the session is Dr. Shyama Prasad Saha. On behalf of the Organizing Committee, I would like to extend a very warm welcome to Dr. Saha. From my bottom of heart and on behalf of Organizing Committee, I truly express my gratitude to you for accepting our invitation to deliver the talk in this workshop. It's a great honor and privilege for me to introduce you, Dr. Saha. Dr. Shyama Prasad Saha is presently working as an assistant professor in the Department of Microbiology, University of North Bengal, West Bengal, India. He is having more than 10 years of teaching experience and has taught several courses at the postgraduate level in the Department of Tea Science, Department of Food Technology, University of North Bengal. He has completed his Ph.D. in Biotechnology with a specialization in Microbiology, Biotechnology and enzymology. His main focus of research has been on the production of microbial xylanase and its role in xylitol production. His current research interest lies on carbohydrate active enzymes, that is Cazymes, antibiotic producing microbes, plant growth promoting bacteria, probiotic bacteria. He has actively participated in both national and international conferences which held in India. He has published his research paper in several high-impact international journals of repute. He has been author of many chapters, contributed to international and national books. Dr. Saha is an active member of Microbiological Society of India. Without taking much time for much awaited talk, with these few words, I would like to invite Dr. Saha to deliver the lecture. Now session is over to Dr. Saha. Thank you.
Hello everyone. Today I am going to start my lectures on measures of dispersion. Myself Dr. Shyama Prasad Saha, Assistant Professor, Department of Microbiology, University of North Bengal. So at first we have to know what is dispersion. The variation of different item of a series from its central value is known as dispersion. That means the scatteredness, variation or deviation of a set of observation from its central value. Now what is central value? Central value is actually mean, median or mode. So from mean, median, mode, the values that are being varied, this is known as the dispersion. For example, if two sets of data may have the same mean but still they may differ in their scatteredness. Just look at this case study. Two students appearing at an interview for the post of lecturer who can teach microbiology, genetics, biochemistry and biostatistics. The marks secured by them out of the 100 are represented below. For example, the student 1 obtained the marks in microbiology 10 out of 100, in genetics 20, biochemistry 80 and biostatistics 90. And student 2 obtained in microbiology 45, in genetics 55, in biochemistry 40 and in biostatistics 60. So the total number of marks they obtained or secured is 200. So the average marks obtained by the student 1 is also 50 and obtained by the student 2 is also 50. So looking at the value of central tendency, in this case it is the mean, we are not able to differentiate the quality of the two students. But if we look carefully into the data, as because the data is very short here, we are able to look into this data in detail. So here we can find that the student 1 is very good in two subjects like biochemistry and biostatistics, whereas he secured a poor marks in microbiology and genetics. But on the other hand, the student 2 obtained an average consistence mark in all the subjects. So here, if we determine the dispersion, then we only able to find the scatteredness of the data or randomness of the data. If we find out any of the measures of dispersion, we can find that the student 1 has a higher scatteredness and student 2 has a lower scattered value. So that's why student 2 is much more consistent in all the subject than that of the student 1. And this comparison is only possible on the basis of the measures of dispersion, not on the basis of the mean. Now, what are the objectives of measures of dispersion? To judge the reliability of measures of central tendency, to make a comparative study of the variability of the two or more series, to identify the cases of variability with a view to control it, and at last to serve a basis for further statistical analysis. Now, what are the measures of dispersion? Actually, 4 measures of dispersions are there. First one is range, then the quartile deviation, third is mean deviation, and fourth is standard deviation. Now, the first one, that is the range. What is range? Range is a measures of dispersion which is based on the two extreme values of series. Range is equal to highest value minus lowest value. That means for determination of the range, which is a measures of dispersion, we have to concentrate on the highest value as well as the lowest value of the data series. Now take an example where the computation of the range of ungrouped data, how to calculate, it has been displayed. Calculate the range of following set of data regarding the number of grains found in 10 panicles. So here is certain numbers of panicles are there, so we need to find out the range. So if we look at the data carefully, so the highest number is of the series is 284 and the lowest value in the series is 76. So that's why the range is 208. It is a very highly dispersed. The data is highly dispersed. That means very low value as well as very high value in the data are there. So it is highly scattered data. So how the range is being calculated from the group data? So group data are those data where the data is represented in terms of its variable as well as in terms of its frequency. So in that such kind of data is known as the group data. So here is the technique to calculate the range of the group data. So you can find that percentage of eosinophils are given in one row and number of person containing this eosinophil count are given in the another row. So this is, we can say it is my variable x and this is the frequency, that is the f. So from here we are able to find that although the highest value is 13 but there is the frequency is 0, that means no person is there that contain the 13% of eosinophil. So that's why the next highest value is 12, because 12 contain 4 number of person that contain the eosinophil. 4 number of person contain 12% of eosinophil. So that's why the highest value is 12, but the lowest value is 7. Whatever the frequency is that, whatever the value of frequency, so the lowest value is 7 and the highest value is 12. So the range is 12 minus 7, that is equal to 5. Now, how to calculate the range when the data is group data but it is being represented in terms of class interval? So here the range is being calculated is equal to upper limit of the highest class minus lower limit of the lowest class. So here the data series is being given where the blood pressures are being represented. Systolic pressure is given in a class interval 71 to 75, 76 to 80, 81 to 85, 86 to 90 and 91 to 95. So here we can find that means the lowest class is 71 to 75 and the highest class, we need to find out the highest class, that is 91 to 95. Now according to the formula, lower limit of the lowest class is 71 and upper limit of the highest class is 95. So the range is calculated as 95 minus 71, that is equal to 24. So these are the very simple calculation, so by which we are able to calculate the range, which is the measures of dispersion. Now what are the merit of range? Range is easily calculated as you have studied and readily understood. Now what is the demerit of range? It is affected greatly by fluctuation of sampling it is not based on all the observation of the series. That means all the observation of the series are not encountered during the calculation of the range. So it is a very demerits of the range. To overcome that, we need to use the another measures of dispersion, that is the quartile deviation. Now what is quartile deviation? It is a measures of dispersion which is based on the two quartile, upper and lower quartile of the series. It is half the difference between the upper quartile, which is represented as Q3, and lower quartile Q1. The difference between these two quartile, that means Q3 minus Q1, is known as interquartile range, and the half of this interquartile range is called semi-interquartile range. So the quartile deviation is also known as the semi-interquartile range. So look at this figure. Here the data is being normally distributed and the data is being splitted into the 4 part and with a 3 quartile, Q1, Q2, Q3. The Q1 represent the 25% of the data, Q2 represent another 25% of the data, and Q3 represent the another 25% data, and so on. So here the distance between this Q1 and Q3 is called Q3 minus Q1, is known as the interquartile range, that is Q. And when it is divided by 2, it is called semi-interquartile range or quartile deviation. That means the formula for calculation of quartile deviation is equal to Q3 minus Q1 divided by 2. So calculate the quartile deviation for the following sets of data showing the blood pressure of 11 persons. So the blood pressure of 11 persons are being represented here. So from here we need to find out the quartile deviation. At first we need to arrange the data in ascending order. So we arrange the data in ascending order. So we need to calculate what is the number of item. So here is the number of item is 11 and the number of item 11 is a odd number of item. So Q1 should have been calculated using this formula. It is n plus 1 by 4 th item. That means the value of 11 plus 1, that is 12 by 4, that is third item is the first quartile, that is third item is 1, 2, 3, 120 is the first quartile, and the third quartile is being calculated using the formula 3 into n plus 1 by 4, as because the total number of item is 11, it is odd, so this formula are being used. So 3 into n plus 1 by 4 th item, that means the value of 3 into 11 plus 1 by 4, that means the ninth item. So ninth item in the series is 128. So Q3 is 128, Q1 is 120. So the quartile deviation is equal to 128 minus 120 divided by 2, that is 8 by 2, that is equal to 4. So in this way the quartile deviation is calculated when the data is ungrouped. Similarly, the quartile deviation also being calculated from the group data when the data is represented in terms of x and frequency. So here is the data. So how to calculate that? So we need to prepare a table at first. So here the number of grain, it is represented as x. Number of panicle, which is frequency, it is represented as f. We need to find out the cumulative frequency. So the first cumulative frequency is 1, then it is added with the 3, it is 4, then this 4 is added with the 5, then it is 9, then 9 is added with the 7, then it is 16. So in that way cumulative frequency are being calculated and the ultimate cumulative frequency is 31. That means we can say the total frequency of this series is 31, which is also a odd number. So as because it is odd number, so the same formula is used for determination of the Q1. It is n plus 1 by n plus 1 by 4 th, that is 31 plus 1 by 4, that is 32 by 4, that is 8 item of the series. 8 item of the series, we need to look at the cumulative frequency table. So 8 falls in this region. There is cumulative frequency is 9 here. So 8 item falls over here. So this corresponds to the 124. So the Q1 is 124. Similarly, we need to find out the Q3, and according to this formula we found it is the 24th item. So we'll again look into the cumulative frequency table. We found here it is 26. So obviously 24th item falls for the Q3 over here in this row, and in this row the value of the x is 130, so Q3 is my 130. Now it is easy to calculate quartile deviation, that is Q3 minus Q1 by 2, that is 130 minus 124 by 2, that is 6 by 2, that is 3. So 3 is the quartile deviation. Now, how to determine the quartile deviation from the group data when it is given with the class interval? So previously also it is the determination of the quartile deviation of the group data, but the data is not presented in the form of the class interval. But when the data is presented in the form of the class interval, so this formula are being used for calculation of the quartile. That is Q1 is being calculated as L1 plus N by 4 minus C1 into I by F1, and Q3 is calculated using this formula L3 plus 3N by 4 minus C3 into I by F3. What is L1? L1 is the lower limit of the class where Q1 falls. L3 lower limit of the class where Q3 falls. C1 cumulative frequency preceding the class where Q1 falls. C3 cumulative frequency preceding the class where Q3 falls. F1 it is the frequency of class where Q1 falls, and F3 it is the frequency of the class where Q3 falls, and I it is the width of the class, and same formula again is used, QD is equal to Q3 minus Q1 by 2. So let's take an example. So in this example, the soil pH and the number of earthworm. The soil pH, which is represented as X, it is being represented in the form of the class interval. So 5.6 to 6, 6.1 to 6.5. So in this way the class intervals are given. So number of earthworm are also presented in this column. So from here we need to find out the cumulative frequency like previous. It is 3, 3 plus 8, 11, 11 plus 9, 20, 20 plus 7, 27, 27 plus 1, 28. So total number of item over here is 28, which is an even number. So Q1 and the width of the class is equal to, that is the difference between this boundary, class boundary, it is the class limit, and the class boundary, the difference between the this class boundary is 0.5, that is I is equal to 0.5. So Q1, as the total number of item is 28, which is an even number, so Q1 it is the n by 4th th item. So look carefully, when it is an odd number, then Q1 is being calculated as n plus 1 by 4. But here, as because it is even number, so Q1 is being calculated as n by 4, that is 7th item. So 7th item, it is false, it is greater than 7 is greater than 3 cumulative frequency table if you look carefully in the cumulative frequency table, we can find that the 7 greater than the 3, so obviously 7 falls here in the, but it is lower than 20, so obviously Q1 falls here. So when Q1 falls here, so it is the quartile frequency class. So L1 is equal to 6.5, F1 is the F1 is equal to 8, it is the frequency of the quartile class, and C1 it is the frequency of the preceding the quartile class, that is the cumulative frequency of the cumulative frequency preceding the quartile class. So that's why C1 is equal to 3. So it is 3. So we get the L1, F1 and C1. Similarly, Q3 is being calculated as 3n by 4. In case of the odd number of item, it is 3n plus 1 by 4. But here, as because it is even number, so it is 3n by 4 only. So it is 21st item, and you can look at the cumulative frequency table. So 21 falls here. So cumulative. So it is the Q3 class. So the lower limit of this Q3 class is 7.1. So L3 is equal to 7.1. Frequency of this quartile class is 7, that is F3 is equal to 7, and C3, which is the cumulative frequency preceding this quartile class, that is 20. So C3 is equal to 20. Now for calculation of the Q1, put all these things in the formula as displayed in the previous slide. So the value is 6.35, and for the Q3, again using the formula that is displayed in the previous slide, so the value of Q3 is obtained, that is 7.171. Now the quartile deviation is being calculated as 7.171 minus 6.35 by 2, that is 0.4105. So now what is the merits of quartile deviation? It is simple to calculate and easy to understand. It is not affected by the values of extreme item, which is being affected by the range, but in case of quartile deviation, it is not affected by the extreme item. Demerits of quartile deviation: it is not based on all the observation of the data, as it ignored the first 25% and last 25% of the item. That means the same problem exists like that of the range. In quartile deviation, we are not considering all the data. It is not capable of further algebraic treatment. It is considerably affected by fluctuations of sampling. Where there is a considerable variation in the values of various items, the quartile deviation is not a suitable measure of dispersion. To overcome this problem, we need to encounter another measure of dispersion, that is mean deviation. Now, what is mean deviation? Mean deviation of a set of observation of a series is the arithmetic mean of all the deviations without their algebraic sign taken from its central value. That means from central value, each of the variable that are being differed, their average or mean is known as the mean deviation. So mean deviation is the sum of the deviation in absolute term from a central value and divided by the number of items in the series. But what should be the central value? The central value may any of the averages, mean, median, and mode. Generally, arithmetic mean is taken as central value. So how to calculate the mean deviation? Here is the formula: summation of within modulus d divided by n, or summation of within modulus f into d divided by n for ungrouped and group data respectively. What is d? D is equal to x minus x bar. X is equal to the values of a particular variable, and x bar is the mean of this series, and n is equal to summation of f. So how to calculate? So using this example, it is very much clear to you how to calculate mean deviation and what is the mean deviation. It is being very much easy to you after looking at this example. So total number of eggs laid by the insect of a species in a single batch were as follows. Calculate the mean deviation from this data. So here the data is being represented. Here is no frequency, so it is ungrouped data. So in this ungrouped data, how to calculate mean deviation? So at first we need to arrange the data in this tabular form. In one column, we need to write down the number of eggs, that is in x, and it is being written. So summation of x is 100, total number of item is 10, so n is equal to 10, summation of x is equal to 100. So what is x bar? X bar is equal to, that is the mean, arithmetic mean. So arithmetic mean is equal to summation of x divided by total number of item n, that is 100 by 10, that is equal to 10. So mean deviation, calculate. To know the mean deviation, we need to find out the d, value of d. So d is equal to x minus x bar. So it is 7 minus 10. 7 is the x and 10 is the x bar. So 7 minus 10, it is minus 3. Then 10 minus 10, it is 0. 13 minus 10 is equal to 3. So that means minus, plus, all signs are there. But we need to omit this minus sign because we need to take the absolute value of the d. That means always positive. So d within modulus d is equal to within modulus x minus x bar. So minus 3 become 3, 0 become 0, 3 become 3, minus 1 become 1. That means we need to take only the absolute value. All are in plus. Negative sign is being omitted over here. And then we need to take the summation of d, that is equal to 14. So if we sum up all this data, so we can find the summation of d, that is equal to 14. And according to the formula, mean deviation is equal to summation of d by n, that is equal to 14 by 10, that is equal to 1.4. So that is the mean deviation of this series. Now, determination of mean deviation from group data. So what is the formula for determination of mean deviation of group data? Here is the formula, that is summation of within modulus f into d by n, where n is equal to summation of f, and f is equal to frequency, and d is equal to x minus x bar. Now take the example. In one investigation, the following results were obtained regarding the number of crustose lichen plant in a hilly slope. Calculate their mean deviation. So here is the data. In the next slide, it is being calculation are being given. So we need to arrange the data in a tabular form. So here is the number of crustose lichen, which is x. So it is 18, 19, 20, and so on. Number of plant, it is 1, 2, etc. That is 18 crustose present in one number of plant, 19 crustose present in two number of plant, 20 crustose present in two number of plant, 21 crustose bearing plants. There is a 5 number of plants that bear the 21 like crustose lichen. So here is f, here is x. Now we need to find out fx. Just multiply 1 into 18, 2 into 19, etc. And then we need to find out the summation of fx. So summation of fx means in this column, if we added all these things, so summation of fx is equal to 314. In this column, we need to find out the summation of f is equal to 15. We need to find out the mean, that is x bar, that is equal to summation of fx by summation of f, that is 314 by 15, that is equal to 20.93. So here, as because it is 20.93, then we need to find out d. We know d is equal to x minus x bar. So it is 18, x is equal to 18 and x bar is equal to 20.93. So it is 18 minus 20.93, that is equal to minus 2.93. Then 19 minus 20.93, it is minus 1.93. Then 20 minus 2.93, that is minus 0.93. So on, we need to calculate this x minus x bar. So in this column, the x minus x bar are being calculated. So it is d, we have f, we need to find out fd. Just multiply f into d. So it is 1 into minus 2.93, that is minus 2.93, 2 into minus 1.93, that is minus 3.86, then 2 into minus 0.93, that is minus 1.86. So in this way, we need to find out the fd. So we can get both the negative sign as well as positive sign, but we need to take the absolute value of the fd, because within modulus we need to calculate the fd. So absolute value, that is negative sign, is being omitted. So it is 2.93, 3.86, 1.86 absolute value we have taken, and sum up all these absolute value. So it is the summation of fd. So summation of fd is equal to 17.35. Now according to the formula of mean deviation, it is summation of fd divided by n, that is the 17.35 by 15, that is equal to 1.1567. So that is the value of mean deviation. So it is the way for calculation of the mean deviation when the data is given in a form of class interval group data, not in the class interval format, sorry. So now, how to determine the mean deviation of group data when it is given with the form of class interval? So here is an example. The distribution of pulses in 1000 tons in Kharif season in 30 districts of Odisha during the period of 1998 to 99 is given below. Calculate their mean deviation. So here the production of pulses in 1000 tons and number of districts are being given there. So you need to calculate the mean deviation. So it is a group data given with class interval. So how to calculate this? Now look at this. We have to prepare the table. So it is the production of pulses, and here is the number of districts. So this is not x. We are not saying that this is the x value. It is the class interval. So 0.51 to 5, the number of districts is 17. 5.1 to 10, the number of districts is 5, and in this way. So we need to calculate the summation of f. That is summation of f is 30. Then we need to find out the class mid value. Class mid value is being calculated as 0.1 plus 5 divided by 2. So the middle value of this class is 2.55. Similarly, the middle value of this class is calculated as 5.1 plus 10 by 2, that is 7.55. So in this way, we need to find out the middle value of the class mid value. Okay, so here, oh sorry, I am written it is x, so it become fm, or you can write down the class mid value as x. So we need to find out the fx. So if this is x, so f into x, we have f, we have x, so just multiply them. So it is 43.35, 5 into 7.55, 37.75, 4 into 12.55, that is 52.5. So in this way, we need to calculate the fx. So here, from here, we can calculate the summation of fx, that is 216.50. So we have summation of fx 216.50. We have summation of 30. So just divide the summation of fx with summation of f. We get the mean value x bar. So that is equal to 7.217. Now we need to find out the d, that is equal to x minus x bar. So my x is 2.55, x bar is 7.217. So 2.55 minus 7.217, it is minus 4.667. Then it is 7.55 minus 7.217, it is 3.333. Then it is 12.55 minus 7.217, it is 5.333. So in this way, we can calculate the d. Again, we need to find out the f into d. That is, we have d, we have our f, just multiply them, we can calculate the fd. So 17 into minus 4.667, it is equal to 79 minus 79.333, then it is 1.667, then 4 into 5.333, it is 21.333. So in this way we can calculate the FD. Once we calculate the FD, we need to find out the absolute value. Again, we are omitting the negative sign. We are taking the absolute value in positive sign, that is 70 minus 79.33 becomes 79.33 only, then 1.667. So in this way we can find out the values of the FD, and then at the very end we need to find out the summation of FD within modulus, that is equal to 158.667. So now the formula for calculation of the mean deviation is same, that is summation of FD by summation of F, that is 158.667 divided by 30, it is equal to 5.288. Okay, so now another term, that is the coefficient of mean deviation. Now what is the coefficient of mean deviation? Coefficient of mean deviation is being calculated when the mean deviation is again divided by the mean. So in this case we have our mean deviation 5.288 and our mean is 7.217. So just divide the mean deviation with the value of mean, we obtain the 0.733. So it is the coefficient of mean deviation. So what are the merits of mean deviation? Mean deviation is rigidly defined and its value is precise and definite. It is based on all observation of the series, unlike quartile deviation. It is not affected very much by the value of extreme item. It is readily understood. Now, what is the demerit of mean deviation? Mean deviation ignores algebraic sign of the deviations and it is not capable of further mathematical treatment. So it is a very much drawback because we are ignoring the algebraic signs. As a mathematics, ignoring the algebraic sign is not so good. So it is a very major drawback of mean deviations. We are ignoring the algebraic sign, that is minus become plus. So algebraic signs are become ignored in mean deviation. It is a very major drawback for mean deviation. So to overcome that, we need to encounter standard deviation. So what is standard deviation? So mean deviation ignore algebraic signs of the deviation. So one of the easiest way of doing away with algebraic sign is to square the figure. If we square the figure, that is if the value is -2, if it is square, that is -2 into -2, so it becomes plus. And again, if it is being rooted, square root, so again the same value is obtained and there is no problem with the sign. So this process is adapted for the calculation of the standard deviation. So standard deviation of a set of observation of a series is the positive square root of the arithmetic mean of the squares of all the deviation from the arithmetic mean. So here is the formula for calculation of the standard deviation. Standard deviation is equal to root over of summation of d square by n. It is the simplest formula, that is root over of summation of d square by n. Or you can write down in this way, that is summation of d square by n, that to the power half, that means the square root. Or we can write down, as because we know that d is equal to x minus x bar, so it is the summation of x minus x bar whole square by n. Or we can write down root over of 1 by n into summation of x square minus summation of x by n whole square. That means if we split this formula, we obtain this formula, that is root over of 1 by n into summation of x square minus summation of x by n whole square. So where x bar is the arithmetic mean, you all know that d is equal to x minus x bar, that is the deviation from the mean, and n is equal to the number of observation. So here is the calculation of the standard deviation from ungrouped data. So here the data is being given. So in the ungrouped data, that means only the data variables is there. It is not represented in the form of frequency. So the number of x are being represented as x. So it is 7, 10 and so on. So we need to find out the summation of x, that is 103. Total number of item is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So total number of item is 10. So we get summation of x. So x bar is equal to summation of x by 10, that is the 10.3. So we obtain the 10.3 is our x bar. So now, like calculation of the mean deviation, we need to calculate the d, that is equal to x minus x bar. So it is 7 minus 10.3, that is equal to minus 3.3. That is 10 minus 10.3, it is minus 0.3, then 10 minus 13 minus 10, that is 2.7. So in that way we need to calculate the values of the D. Now in mean deviation we are taking the absolute value of the D, that is minus 3.3 become 3.3, but here we need to find out the D square. Just square them, that is minus 3.3 into minus 3.3, that is equal to 10.89, and minus into minus become plus, it is 10.89. so it is mathematically correct to omit the negative sign. So similarly in that way all the D become converted into D square. So here we find our D square, that is D square is equal to 44.1. so the standard deviation is equal to root over of summation of D square by n, that is root over of 44.1 by 10, that is root over of 4.41, that is equal to 2.1. That is the answer, and that is the way of calculation of the standard deviation from the ungrouped data, where the problem of omitting the ignoring the negative sign is being solved, because we are squaring over here and then we root over the thing again. So in that way the value of standard deviation is obtained here, it is 2.1. Now determination of SD from group data. So we need to use this formula, that is SD is equal to root over of summation of FD square by n, that is root over of summation of f into x minus x bar, because we know that D is equal to x minus x bar. So x minus D square means x minus x bar whole square by n, or if we split these things we can obtain, that is root over of 1 by n into summation of fx square minus summation of fx by n whole square. What is n? Capital N, it is summation of f, x bar is equal to mean, and D is equal to x minus x bar. So here the data is represented in the form of the frequency and the values, x and f are there, so it is a group data. So here is the data. So how to calculate the standard deviation? So we need to prepare the table again. So we need to find out the number of x, we need to put the values of the x, that is number of x, here is the values, number of insect is 2, that is 7 number of eggs laid by the 2 number eggs, the two number of insects. Similarly 3 insects are there which can lay 8 number of eggs, 3 insects are there which can lay 9 number of eggs, similarly 4 insects are there which can lay 11 number of eggs, and so on. So here the data is presented in the form of the x and frequency f. So we need to find out the fx, because we need to find out the mean, so we need to find out the fx again. So 2 into 7, that is 24, that's 14, 2 into 7, that is 14, 8 into 3, that is 24, 9 into 3, that is 27. So in this way we can find out our value of fx. So now we need to find out the summation of fx. Just added all these things, so summation of fx become 203, and summation of f, just added this row column, that is summation of f is equal to 20. So x bar is equal to summation of fx by summation of f, that is 203 by 20, that is 10.15. Now we need to calculate the D, that is equal to x minus x bar. Now how to calculate this D? We know the x, that is 7, so 7 minus x bar, that is 10.5. So 7 minus 10.5 it is equal to minus 3.15. 8 minus 10.5, that is minus 2.15. So in this way we are calculating the values of the D over here. So once we obtain the D, values of the D, just square it, that is D square. Okay, that is minus 3.15 into minus 3.15, that is 9.12, minus 2.15 into minus 2.15, that is 4.62. So in that way we are calculating the values of the D square over here. Then we need to find out the f into D square. So we have the values of f, we have the D square value, just multiply it. 2 into 9.292, that is 19.84, 3 into 4.62, that is 13.86. So in this way we are calculating the values of f into D square. So summation of fd square is equal to 78.55. Just added these values of individual fd square, so it is summation of fd square is equal to 78.55. So according to the formula, SD is equal to root over of summation of fd square by summation of f. So summation of fd square, the value is 78.55, that is divided by 20. So the values obtained is 1.98. So in this way the standard deviation is calculated from the ungrouped group data. Now, determination of standard deviation from the group data given with class interval. The same formula for the determination of the standard deviation from the group data is used. That is, standard deviation is equal to root over of 1 by n into summation of fx square minus summation of fx by n whole square. So here the amount of the rice production in 1000 ton during the period of 1998 to 1990 in 30 districts of certain states in India is given below. So here is the class interval, here is the frequency. There is rice production is in the form of the class interval and number of districts in the form of the frequency are represented here. So how to calculate these things? So rice production and number of districts are there. So number of districts is 5. So here from here we need to find out the summation of f. So summation of f is equal to 13. Now we need to again find out the class mid value, as we have already find it out from the mean deviation. So it is the middle value 0.1 plus 1 by 2, that is the middle value of this class. 0.1 plus 1 by 2, that is 0.55. Similarly 1.1 plus 2 by 2, that is 1.55. So in that way we are obtaining the class mid value. So once we obtain the class mid value, just treat these things as a value of x. So now we need to find out the fx. So 5 into 0.55, that is 2.75. 7 into 1.55, that is 10.85. So in this way we are calculating the values of the fx. Now just added all these fx, individual fx, we can find out the summation of fx, that is 106.5. So according to the formula for determination of the mean, x bar is equal to summation of fx. So we need not to find out the x bar over here, because we are using the separate formula. So we just find out the f into x square, f into x square. So x is there, so f is there, so 5 into 0.5 whole square, that is 1.51. Similarly 7 into 1.55 whole square, that is 16.81. So in that way we can find out the fx square. So we can find out the summation of fx square, that is 558.07. So the formula for SD is root over of 1 by n into summation of fx square minus summation of fx by n whole square. So root over of 1 by 30, that is n is equal to 30. So 1 by root over of 1 by 30 into summation of fx square is equal to 558.07. So just write it down, 558.07 minus summation of fx. The summation of fx value is 106.5. So 106.5, n, the value of n is 30, that is whole square. So if we do the math, we can find out, that is easy to calculate. We can find the 2.4495. That is the value of the standard deviation. So once we find out the standard deviation, we are interested also to find it out the standard error. So now what is standard error? Standard error is easily calculated when the standard deviation is divided by root over of n, that is the number of sample. So in this case the standard error is being calculated as the value of standard deviation, that is 2.4495 divided by root over of 30, that is 2.4495 divided by 5.47, that is 0.44. So on the basis of this standard deviation and standard error, we are able to judge the consistency of the data, whether the data is so much scattered or not. If the value of standard deviation is so high or the value of standard error is so high, then we can say that the data is highly scattered or randomness in the data is very high. If the values of the standard deviation or error is very low, then the values is much more consistent than the previous. So in this way the standard deviation and standard error are being calculated. So what are the merits of standard deviation? So standard deviation is rigidly defined and its value is always definite. It is based on all the observation of the data. So that is the very good point or very good merit of the standard deviation. It is amenable to further algebraic treatment. It is less affected by the fluctuations of the sampling. Now what are the demerit of standard deviation? Standard deviation is not easy to calculate, because we know that there is a very complex formulas are there. You need to remember those formula for calculation of the standard deviation. It gives more weightage to extreme item and less to those which are near the mean. I hope you will understand that how to calculate the measures of dispersion. Thank you.